in the final stage of translation is termination. So the first stage was the amino acylation of tRNA. The second stage was initiation. Third was elongation. Now it is termination, which essentially means we have created our peptide chain, and now we want to cleave it off from that ribosomal unit. So if this is our 50S subunit and this is our 30S subunit, and the mRNA is over here, when the mRNA, the code in mRNA is written in the 5' to 3' direction. So the code is read from the 5' to 3' direction, and each codon represents 3 nucleotides. And when we reach a stop codon, that is going to signal termination. So there are 3 stop codons that we are most familiar with, and that is U, Ga, Ga in opposite directions. So if we reach a stop codon that says UAG, UAG is one of our stop codons. So the ribosome is going to stall this for a bit and until a release factor comes in. This release factor is RF1. RF1 is going to bind to the A site of the 30S subunit. When it binds to that A site, it's important to note first of all that we have 2 release factors. We have RF1 and RF2. RF1 will bind to UAA or UAG, and RF2 will bind to UAA or UGA. In this case, since we have UAG, RF1 is the one that's going to be binding to that A site. Once RF1 binds, it's going to facilitate the cleavage of this peptide that we have created from this mRNA, and it's going to be cleaved off. But now the next part is that now that we have this RF1 that's bound to that A site, we have to remove it. So we have to remove this, and how do we remove this? We need another release factor, and this is release factor 3, which is responsible for removing both release factor 1 and 2, depending on which we have. So RF3 will come in, and it will remove that RF1. But now we also have to get rid of the release factor 3. What gets rid of the release factor 3? It's another factor called the EF factor G, which has a GTP bound to it. So in order to essentially, when our release factor binds, another release factor comes in to remove the previous release factor. But in order to remove the other release factor, we need this other factor that is EF, and it has that GTP bound to it. And the hydrolysis of GTP essentially facilitates this reaction, that energy release. So now we have EF, that factor bound with GDP, and we lost an organic phosphate. As a result, when this entire process occurs where the release factor 3 removes that release factor, the release factor 3 is removed by EFG. As a result, the other components also leave. So this free tRNA. This free tRNA will also be free to leave, because right over here, when we cleave that peptide, we're cleaving it in the peptidyl site, so it's in the P site, from that tRNA, which is carrying that peptide, but the tRNA is still left over. And after the EFG and the hydrolysis of GTP and the release factor 3, and when the release factor 2, everything is gone, the tRNA will also leave, and that mRNA will also dissociate. Once everything is removed, our IF3, so our initiation factor number 3, is back, and it's going to bind to that E site. It's going to bind to that E site because it's going to prevent the binding of the 50S subunit. Now, once we are at this stage where IF3 is bound to the E site, eventually IF1, initiation factor number 1, will come and bind to the A site, and we will undergo another cycle starting from initiation, elongation, and then termination to yield another peptide. So essentially, the termination step RF1 or RF2 will bind. RF3 will remove RF1 and RF2. EFG removes RF3. GTP is hydrolyzed. Everything is off of the ribosome. Then IF3 comes and binds to prevent that binding with the 50S subunit. So we're ready to go for another round of synthesizing that peptide. So overall, termination utilizes one GTP equivalent to cleave that peptide off.